A very warm welcome in the Life Science Academy. Today we will learn cell composition and architecture of plasma membrane. So what is the constituent and what is the plasma membrane? Prior going to discuss about the plasma membrane, we have to a term known as the cell. What is the cell? What is the cell? We will simply define the cell. Cell is the structural and functional unit. What we call it? A structural and functional unit of life. Without cell, we can't imagine the life came on earth. So cell is the basic unit of life. Uh, Every organism possesses cell and its constituents. Today we will learn composition of plasma membrane. Plasma membrane is a part of a cell. So we have a typical structure of cell. This is the cell and this is the plasma membrane. The boundary is defined as a plasma membrane. So here we have a first property of plasma membrane, that is it defines the boundary of a cell. It defines the boundary of a cell. Under the plasma membrane, there will be the presence of cytosol. There will be presence of cytosol. So this is the cytosol. Under the cytosol, there will be presence of nucleoid in case of prokaryotic cell and nucleus in case of eukaryotic cell. So this is the basic composition of cell. So every organism possesses three things common. First one is the plasma membrane, second one is the cytosol, and third one is the what? Genetic material. Whether the genetic material either RNA or DNA. So we will learn detail about the plasma membrane in this lesson. So we have some physical properties which define the plasma membrane. So the physical properties included that flexibility. Plasma membrane have a flexible in nature and a self-sealing capacity and selectively permeable to polar solutes. So we have some terms like polar solutes. So what is the difference between the polar and non-polar things? We have a basic idea about the polar and non-polar. Can you say that plasma membrane is non-polar in nature? Why it is not polar? Why it is not polar? Why it is non-polar? We have a typical structure. This typical structure is known as the head, and this is the tail. The whole structure is defined as a lipid molecule, which possesses head and tail region. In the tail region, there will be presence of some alcohol group and or bulky sugars, and there in head region there will be presence of fatty acyl chain. So this typical structure, and in head region there will be some charge also present. It may be negative or it may be positive, like acetyl, like phosphatidylserine, like phosphatidylserine, and phosphatidylethanolamines. Some phospholipids have a charged. So in that head region there will be presence of charge also possesses. So as you all know that lipid bilayer is a non-polar in nature. So that's why it creates a boundary. It creates a boundary. So in the surrounding region, there will be presence of what? Water. In the surrounding, there will be presence of water. And apart from surrounding, in the or you can say under the cytosol, there will be presence of also a water. So outer and inner area is rich in water molecule, and lipid bilayer is a non-polar and water is a polar molecule. Water is a polar molecule, and polar molecule have some charge, some partial charges present in the polar molecule. In non-polar molecule, there will be no presence of charge, no charge. So partial charges present in the water molecule and due to the presence of hydrogen oxygen, and non-polar molecule is not charged. So if we want to interact this water from this water, what to do? We have to do that. First of all, there will be presence of what? Plasma membrane. This plasma membrane is a barrier for polar solutes and solvents. And universal solvent is a water. So water is diffuses from the plasma membrane, but under the very low concentration. We have some water channels. We have some water channels. What we call it as a aquaporins. In the aquaporine regions, water is rapidly enter and out from the lipid bilayer. So why we call it as a flexible in nature? The cell is plasma membrane is flexible. Why? Because in the plasma membrane there will be lots of concentration of protein and lipids molecule, and this protein and lipid molecule will be vary in the thickness. So cell membrane is sometimes thicker and sometimes thinner in the particular region, or you can say area. Why we call it a self-sealing capacity? So as you cut this region, as you cut this region, the cell membrane is spontaneously, which we call it say spontaneously reseal again after some time, and selectively permeable to polar solutes, and selectively permeable to polar solutes. Sometimes this is the plasma membrane. There will be presence of glucose molecule. So what is the glucose? Glucose is a sugar and is a solute. So solute is cross are not in the plasma membrane. As you all know that polar solutes cannot cross. So we have some carrier proteins. We have some carrier protein for the transportation of glucose, and water is diffuser is across via the diffusion, via the diffusion, but not fast. But gaseous molecules like oxygen, carbon dioxide, or ammonia, these gases are easily cross the plasma membrane. Why? Because it also are non-polar in nature. So we simply said that non-polar and polar molecules interact interact each other via the hydrophobic interactions. Via the hydrophobic interaction, hydrophobia occurs when there will be presence of what? Water. If no water, no hydrophobia. No hydrophobia, no hydrophobic interaction. So uh, polar and non-polar molecule interact via the hydrophobic interactions. If we're talking about the major constituents of plasma membrane in various organisms, so it varies in every organism. Its concentration is vary in every organism. Uh, if we're talking about the major components is the protein and the lipid and also carbohydrate. But remember, carbohydrate is not present in free form. It is conjugated either the protein molecule or the lipid molecule. So, if we're talking about human myelin sheath, which is rich in protein 30% and phosphorate 30%. approximate equal weightage of both protein and lipid moieties, and somewhere sterol is also present. This sterol is known as a cholesterol, and other glycolipids are also present like galactolipids. So every organism possesses variation concentration in phospholipid, and every single organism may vary a composition of the plasma membrane in different type of cell, in different type of organism, and in different type of organ system. Like mouse liver, it constitutes 45% protein and phospholipid 27%. Here, phospholipid is the major constituent as compared to lipid moiety, and sterol is 25%. And this sterol is a cholesterol molecule. So, depending upon the organism, organ system, and tissue, whether the requirement of lipid, whether the requirement of protein, or whether the requirement of sterol, these three things define the actual property of the plasma membrane and actual organism. If you're talking about yeast have a 52% protein and negligible amount in phospholipids. Why? And sterol is also 4%. And in case of yeast, there will be presence of ergosterol and sometimes triglycerols also present. If we talk about paramecium. So here 56% protein and 40% here है आपके पास lipids and 4% sterol and stigmasterol also present. Here sterol is present but vary in structure and concentration. If you're talking about model organism, which is the E. coli, here 75% protein in its lipid bilayer. Why we cannot say that it is a protein bilayer? Still, we have to say that it is a lipid bilayer. If you're talking about percentage of phospholipids, here we have a 25, only 25% lipid moiety in the E. coli's plasma membrane and no sterol present. If we're talking about E. coli, which is an enteric bacteria, which is a enteric gram-negative bacteria, which resides in the small intestine of human. So this is the typical structure of oval in shape. We're talking about shape and size. So it is oval in shape. E. coli is a oval in shape. So we have a plasma membrane. Here in the plasma membrane where percentage of protein moiety is greater than phospholipids. Why? Because in E. coli possesses lots of transporter proteins in their plasma membrane. And this is the cytosol, and here is a nucleoid, which we call it a genome of E. coli. So this is the basic composition of different organisms. Some may have prokaryotic and some may have eukaryotic. If we're talking about prokaryotes, so E. coli is a prokaryotic organism, model organism. And if you're talking about the model organism of eukaryotic cell, so this is the yeast. Yeast is the model organism in case of eukaryotic cell. So here we have a typical representation of what? The plasma membrane. So this is the fluid mosaic model. This is what? This is the fluid. This is what? This is the fluid mosaic model of plasma membrane, fluid. If we want to represent what? Fluid plasma membrane. So we have some mosaic model. So we have some fluid mosaic model. So this is the fluid mosaic model. What does said that fluid mosaic model? If we called this is the lipid bilayer. This is the lipid bilayer. So we previously said that this is the head and this is the tail part. So here we have head and tail part, and some may have some kink and some may have this like structure. This is the kink, and this kink define the double bonds, double bonds in petiosyl chain, and cis double bond. This is the configuration of cis. We have two type of configuration. One have the trans and one have the cis. So here double bond is present in the cis configuration. So this is define the this structure define the this can define the double bond, denoted as a here double bond is present. So if you're talking about the thickness of the plasma membrane, it is 3 to 5 Armstrong in thickness, or you can say 3 to 50, 30 to 50 nanometer in thickness. What is this region? This region is what? This is what? This is the transmembrane protein. This is what? This is the transmembrane protein, which span the protein, which span the lipid bilayer. Some proteins have one single pass transmembrane domain, and some have multipass transmembrane domain. And some protein only present on the periphery region, only present in the periphery region, like this and this. This is the peripheral protein. This is the peripheral protein, which is present in the periphery of the organism, periphery of the lipid bilayer. So this is the cytosolic periphery. This is the cytosolic peripheral protein, and this is the exoplasmic. This is the exoplasmic peripheral protein, and this is the sugar moiety. This is the sugar moiety. So this is what? This is glycosylation in lipid bilayer. So this is the multipass. This is what? This is the multipass transmembrane protein. What is the function of multipass transmembrane protein? Multipass transmembrane protein form a channel for transporting the some polar solute, some nutrient from the surrounding, and this region, this is the carboxylic region, and this is the amino region, amino part. So if we're talking about only this region, this is the non-polar. This is what? This is non-polar region, and this is polar region, and this is polar region. So what we want to say that here is also presence of water, and here is also presence of water. Surrounding is what? Here surrounding is, in surrounding there will be lots of water is present. But why? Water is not easily across the plasma membrane. We have some physical properties which we call it as a flexible in nature. Cell membrane is flexible in nature. Cell membrane has a physical property which is the flexibility, and second is the self-sealing capacity. So if we want to remove this peripheral protein from the plasma membrane, we have to treat with the salt concentration. If we increase the amount of salt, the plasma membrane and peripheral protein will dissociate from each other, and it's electrostatic. This moiety is this region is attaches via to each other. It is the where is the electrostatic interaction, and this is the electrostatic interaction. This electrostatic interaction is disturbed by the salt treatment. If we want to remove this transmembrane protein from the plasma membrane, it is not easy to detaches from the plasma membrane. Here we use detergents and some type anion and cationic detergents, not simply the salt concentration, to increase the salt concentration. So we have some methodologies for dissociating the transmembrane protein from the plasma membrane. And this is the most interesting part of the plasma membrane. So we have some difference between the positive and gram negative and gram positive bacteria. So on the basis of plasma membrane, we have to divide the bacteria into two different categories. One category is the gram positive bacteria and one is the gram negative bacteria. So what do you say that E. coli is what? E. coli is what? E. coli is the gram negative bacteria. So first of all, we will have to discuss about the gram positive bacteria. So uh, in gram positive bacteria, in the upper region, we have some solid layer of protein. We have some solid layers. And this peptidoglycan, in the middle region, there will be presence of peptidoglycan. In the peptidoglycan region, as you all know that what is present between that NAM and NAG is present, and acetylmuramic acid and acetyl glucosamine. And this is cross bridges between each other via the peptidoglycase, So in the inner region, there will be presence of what? Lipid bilayer. In the lipid bilayer, we have some lipoproteins. We have some lipoproteins. This is the lipoproteins, and this is what? This is the polysaccharide, and this polysaccharides either attaches with the solid layer of the gram-positive bacteria or with the peptidoglycan. So this is the basic and typical structure of the gram-positive bacteria. We have to compare with the gram-negative bacteria. So if what is the difference between those? Between those, between that gram-positive and gram-negative, we have some basic difference. First difference is we have the outer and inner plasma membrane. Here, two layers of plasma membrane will be present in the gram-negative bacteria, and here only one plasma membrane is present. So this is the outer plasma membrane, this is the inner plasma membrane, and between the inner and outer plasma membrane, we have some peptidoglycan layer. But this peptidoglycan layer is very thin as compared to peptidoglycan layer, which is present in the gram-positive bacteria, which is very thick as compared to gram-negative bacteria. So this is the lipoprotein. Lipoprotein is also present in this region. And here some here some porine channels present in the gram-negative bacteria. The porine channel is absent in the gram-positive bacteria. And this is the lipopolysaccharide. This lipopolysaccharide is absent in case of gram-positive bacteria. This is the basic and very beautiful knowledge which define the bacteria into two different categories. And some people know what is the difference between the gram-positive and gram-negative bacteria. So we proceed. Types of lipid aggregate. What is the types of lipid aggregate? When we form micelle, when we form bilayer, when we form vesicle. So if the structure like this, this will form the micelle. This is structure is what we call it as a micelle. Example, the best known example is the detergent. Detergent will form the micelle. And if we talk lipid bilayer, this structure is a bilayer. And if we want to set that what is vesicle, so we have a vesicle. This is the if the lipid bilayer and joining is proceed is spontaneously. This ends were joined by spontaneously. This will form a what is this will form the vesicle. So this lipid bilayer is at the end of this is form the vesicle like a structure, and this vesicle is very important in physiology and physiological condition. So integral membrane proteins, this is the integral membrane, which is also said that which is the transmembrane protein. So alpha helix is the example of integral membrane, or you can say transmembrane protein. What is the work behind the transmembrane protein? This work, it is a recognition or it is a receptor. It worked as a recognition or it worked as a receptor. This is the helical bundle. So this is the multipass transmembrane protein. Uh, very known example is the GPCR. Very known example is the GPCR, which is the seven times span the lipid bilayer. If we want to know that this region is a highly non-polar, so in the highly non-polar region, there will be presence only hydrophobic amino acid, only hydrophobic amino acid. So give the name of hydrophobic amino acid in the comment section. So, and this region, facing the cytosol and exoplasmic phase. So here is presence of water moiety and here is presence of also water moieties. In this region, no water molecule present. In this region, no water molecule present. So strictly, this region is only allowed for hydrophobic amino acid. Approximately 20 to 30 amino acid will span this region of transmembrane protein. So this this application applied all those of that transmembrane protein, and this is the beta barrel. This is the beta barrel. In the beta barrel, there will be presence of the beta sheets, not the alpha helix. Mostly transmembrane proteins are the alpha helical, but some have exceptions like beta barrels. And this is the structure. What does look like that? This is look like as a enzyme. So enzyme is a enzyme is a also a protein. As you all know that all proteins, all enzymes are protein, but not all proteins are enzymes. Some lipids have linked with the proteins. Some lipids linked with the protein attached to lipid bilayer by the covalent bonding. So जो interactions हैं कहाँ lipid bilayer में whether the covalent bonding present, where the electrostatic bonding present in lipid bilayer, some typical chemical bonding represent like electrostatic interaction, hydrophobic interaction, hydrogen bonding, hydrogen bonding, and fourth will be van der Waal bonding, and fifth one is the covalent bond. So write down in the comment section where the electrostatic interaction present, where the hydrophobic interaction present, where the hydrogen bonding, and where the van der Waal present, where the covalent. So as you all know that covalent bond present between the lipid linked proteins, between the lipid linked proteins. आगे के लेक्चर में हम देखेंगे क्या सेल मेम्ब्रेन का कंपोजिशन क्या है, लिपिड के बारे में डिटेल में चर्चा करेंगे। डिटेल कितना टाइप लिपिड कितने टाइप होते हैं लिपिड्स के, कौन ग्लिसरॉल बेस्ड है, कौन स्फिंगोसीन बेस्ड है, और प्रोटीन क्या होते हैं, प्रोटीन के टाइप्स क्या होते हैं, और कार्बोहाइड्रेट किस तरीके से अरेंजमेंट देखा जाता है किसमें, लिपिड बायलेयर में। तब तक के लिए थैंक यू।